Hi, I'm Dr. Ebony Glover, and I am an associate professor of neuroscience at Kennesaw State University within the Department of Psychological Science. I am also the principal investigator of the Affective Neuroscience Lab, which is a research and training program that I developed at KSU to examine a range of biological and psychological factors that may contribute to how we regulate our emotions. I want to talk specifically about my research concerning the role of sex, so whether or not you're male or female, and race, so what racial group do you identify with, and how these factors relate to how you process sensory information that's perceived as threatening and how you respond to that perceived threat via your fear reactivity. And I also want to talk about what drives my research. How did I come to study these particular research questions, and how did I come to use the tools that I use to examine these questions? And so it sort of began very early in my research career when I was an undergraduate student at Spelman College in Atlanta, Georgia. I don't recall the class, but I do recall the very disturbing statistic concerning mental health disparities. So mental health disparities being the disproportionate incidence of certain mental illnesses in certain populations. And what struck me was that women have 2 times the rate of anxiety disorders compared to men. And this is true of a range of anxiety and threat trauma-related disorders, that women are 2 times more likely to have twice the rates of these disorders compared to men. And this just stuck with me, this idea that these disparities existed and that there was a very little research sort of surrounding the underlying mechanisms that would cause such a disparity between the sexes. Later, if we fast forward to, I'm now at KSU, I have my own research lab, and I began to think about the increased awareness of police aggression toward unarmed Black individuals. And I started to think about if there is a way to measure at the very basic neuroscience level how individuals perceived members of their outgroup in terms of how threatening they are. And when I say outgroup, I mean a member of a racial group that is different from your own. And whether or not your reactivity, your fear response, related to whether or not the perceived threat was a member of your own group or a member of an outgroup. And as I thought about these questions, I came to the realization that I actually have the research tools, I have the institutional resources, and I have the expertise and background to actually address these questions, which are complicated social issues, but at a very basic neuroscience level. And so that's what I set out to do in the Affective Neuroscience Lab at KSU. So I want to talk about sort of the background, like how I got to use these tools that I use to study fear, learning, and memory. And it all began, again, when I was an undergraduate student at Spelman College. This individual here, his name is Michael Davis. He came and gave a talk at Spelman, and I was in the audience, and I was just fascinated by his research. So at the time, he was a distinguished professor at Emory University and a pioneer in the neurobiology of fear and anxiety research. And he used rodent models to study the neuroscience of fear. And so after his talk was over, I eagerly approached him and asked him more questions, and ultimately he invited me to come to his lab and gain some research experience. And so, of course, I did. I went to Emory almost daily. I commuted to Emory from Spelman, and I worked in his lab as an undergraduate student. And this experience was just invaluable, and it led to me pursuing a PhD in neuroscience and animal behavior at Emory University, with Michael Davis serving as my graduate advisor. And so here I am as a graduate student working in his lab, holding a rat. And here is the paradigm, the setup that we used, which is a fear conditioning model where we would train rats to fear neutral stimuli, just regular everyday stimuli like a light or a sound or a smell, by pairing that neutral stimulus with an aversive stimulus, in this case, a foot shock. So the animal is in this chamber, they get a foot shock while they're being presented with the stimulus. And ultimately they begin to learn to fear this previously neutral stimulus, even in the absence of the foot shock. So that's fear conditioning. And so Dr. Davis pioneered the measure of startle, which is a motor reflex to a loud noise. You hear a loud noise, you startle. Well, he mapped out the neural circuitry of the startle response as a measure of fear in rats. And I bring all this up because I wanted to show you how very early I began to think about sex differences. And so I saw very early all of the rats we studied were males. And I asked Dr. Davis, why do we only study males? And to summarize it, he told me that female rats are very difficult to study because of their estrous cycle, which is similar to the female, to the menstrual cycle in women, such that depending on where they fell in the estrous cycle across time, they had a hormonal fluctuation and their behavior would be variable. And so to eliminate that, we just studied males because their behavior was consistent across time. And if we wanted to study females, you had to go through this process of removing the ovaries so that you can sort of stabilize their behavior. And so needless to say, it was a bit complex to study females with this mindset that was pretty prominent in the field, that the female subject was just sort of difficult. And so the literature was actually lacking on examining sex differences in fear responding and fear reactivity, even in the face of this well-known literature that women had twice the rates of anxiety disorders compared to men. So I didn't get to study female rats as a graduate student, but I later on pursued looking at male versus the female brain in humans during my postdoctoral years. So to fast forward, when you get a PhD in a field like neuroscience, it's highly recommended if you want to get your own research lab and you want to get funding from major funding institutions, it's highly recommended that you get postdoctoral training, which is additional training in another research lab where you can develop your expertise, you can publish more findings, you can become more competitive when it comes to applying for grants. And so that's what I did at Emory University in the psychiatry department. I did postdoctoral training. But what was so exciting was that I worked with a team that translated the startle, the animal model of startle, to humans. So translational research is when you can take sort of an animal model and then sort of apply it to a human model. So we measured startle in humans as a measure of fear reactivity. And with that paradigm in place, I was able to examine sex differences and specifically look at some biological factors related to sex. So I found that estrogen, which is a sex hormone, a reproductive hormone, was really important when it relates to women and anxiety. And that women with low estrogen levels had heightened startle. And specifically women with post-traumatic stress disorder, if their estrogens were low, their startle response was high. And when their estrogen levels were high, their startle response was sort of normal and neutralized, similar to men. I don't want to call it normal, but it was similar to men. So estrogen seems to be a really important factor in women's susceptibility to anxiety. So I kind of was able to address some of the early questions that I had concerning sex differences and mental health disparities, right? And that was my that was my postdoctoral research. And so when I transitioned from a postdoc to an assistant professor at Kennesaw, I wanted to replicate the paradigm in a healthy sample. Whereas before I was studying a clinical sample, people with PTSD, I came to Kennesaw and I wanted to study this in a healthy population. So I replicated the setup. So this is our setup where we present participants, we put them in a booth, we present them with a neutral stimuli, we measure their startle. So what you see here, and I'll go back, we measure startle via the eye blink response. So we measure the muscle reactivity. When you startle, you blink as well, and we can quantify your startle reactivity by measuring that blink response. So you can see that here as well. And then we quantify it, and this is just sort of an image capture of one of our tools to quantify the blink response. And this is what our setup looks like at KSU. This is what our setup looked like at Emory. And so we have our startle booth. So there's a behavioral room that we have at Kennesaw. And here are some images of some of my fantastic students who are training and are learning how to set up a participant and set up the electrodes to quantify their physiological responses. And here's another student who is looking in real time as we're training someone at their physiology. So you're seeing their blink response here. And you can also see on the computer screen, this is a sort of a replication of what the participant is seeing while they're in the booth. So in this particular study, we use shapes as our sort of neutral stimulus. So it would be like a circle or a triangle, in this case a square. But I also want to add that we later on began to look at faces. So we presented participants with either a face of a male who would be identified as a black male or a white male. So participants saw either a face of a black male or a white male, and we measured their startle responses when we conditioned them to either of those faces. And we also measured some biological factors such as estrogen levels, also progesterone, which is another reproductive hormone, as well as cortisol, which is a stress hormone. And here's a picture of some amazing students at KSU who are assaying some saliva. And so we got an enormous amount of findings from this work, and I just wanted to give you a brief snapshot of some of our findings. One is that when you measure startle to a threatening stimulus, so we pair the stimulus with an aversive stimulus, most times people are going to startle higher to that threat compared to a safe condition. Women who are naturally cycling, so who are not using any type of hormonal contraceptives, startle to the threatening stimulus much higher than males. And we also found that women who were using hormonal contraceptives startled even higher to the threat than naturally cycling women. And so concluding that using hormonal contraceptives and being female, being a woman, increased your amygdala activation and increased your fear responsivity compared to males. We also examined startle response to a outgroup. And so in this particular graph, we're showing the people's startle response in the presence of a black male face. So the black participants are actually looking at an in-group, and the white participants are looking at an out-group. And what we are seeing is that when white individuals see a black male face, they startle higher than a black individual who sees a black male face. And when you present the face over and over again over time, so this is a time point, eventually their startle begins to diminish. That's called extinction. But white participants had a slower extinction to the picture of a black male face compared to black individuals. And again, this is a really brief snapshot. Because I'm almost out of time, I just want to wrap up and just show you some of the amazing just pictures of us traveling across the country, presenting our findings. We just had so many fantastic students come through the Affective Neuroscience Lab and present. This is just I have so many pictures. And I also just want to encourage you as an undergraduate student or a graduate student to continue, if you have not already, to pursue research. It is invaluable. Continue to get those opportunities, and I really hope you enjoyed my presentation, and thank you for watching this to the end.